So now we are on to photosynthesis. And I know the students at my high school should know this very, very well. Just saying that because I taught I taught this lesson to those classes. Um, but to review, and for anybody else that's watching this, uh, there's a lot of detail in photosynthesis. I love photosynthesis because the details are actually pretty amusing and kind of beautiful how everything works together. But you don't need to, this is not going to be a detail focused exam, as I've said many times. You really want to understand the big picture. So, looking again at our three things, our structure and function, our vulnerabilities, those critical steps, and how this process is regulated, right? And there's so the first things, right, in terms of regulation, especially for these processes, how do you regulate it? Well, you have this H2O and this CO2 that comes in to the cell, right? Cell can't really control the light. But if they want photosynthesis to happen, you can regulate intake of CO2, right? And you can do that. The cell plants breathe, right? In a way, they have the stoma that not to be confused with this stroma, but they have those stoma that look like sort of like that, where they've got like these cells, right? That can open or close, right? So it can either open or close, and to either let in air or not let in air, right? So that's one way, the stoma. Again, you probably don't need to know these, just understanding the idea of regulating this process with the intake of CO2. Uh, yeah, so then there are some intermediate steps. Again, you don't need to know all of those. You should know the main ideas behind the light reactions and behind the Kelvin cycle. Uh, the light reactions is going to happen in these thylakoids. And the important thing you're going to want to know is the pancake structure. I just spelled pancake wrong. Um, Pancake. Why do we have the pancake? Okay, so why do we have the pancake? And that's going to be because we want a high surface area to volume ratio, right? Just like the folds in the mitochondria, right? We want to have a high surface area to volume ratio, right? Again, they're not called pancakes, they're called thylakoids, but they look like pancakes, and because you want to have that high surface area to volume ratio, because you're going to have the light reactions happen in the thylakoid membrane, right, with these membrane-bound proteins. Now, if you want to disrupt this process, right, where's some great places to disrupt this process? In these proteins, the photosystem, really where I would think I would want to disrupt this process if you could, is these transport proteins, right? Because if you can't transport the electrons from one place to the next, this whole system fails. So these, right, these might be good places to disrupt. Right, those proteins might be some really good places to disrupt the process if you like denature those proteins, because then the electrons can't move from one place to another, right? um if you affect the ATP synthase, again, this is not a huge part of it, right? The creation, this will, it will definitely make it make the energy of the cell go down. It will make photosynthesis be less, right? Because it won't can't produce ATP in the light reactions. It will less energy helpful, but it won't, right? What's important is the flow of the electrons, right? Again, this is, you have the NADPH, which contains those electrons, right? The NADPH, which contains those electrons, that's what you really want flowing between these processes, right? So it's going to be important for the AP a lot of the times they'll ask you, if you disrupt this step, what will happen to the process, right? So the main places, right, if you disrupt these carriers, that will pretty much ruin the whole process. Um, the other thing you could do, right, if you were looking at sort of those critical steps, is affect the Kelvin cycle, right? And again, the Kelvin cycle, you may want to know sort of the main ideas between these steps. Um, just the Kelvin cycle, what it does is it takes carbon dioxide and turns it into um, organic molecules, right? There's going to be proteins here. This Rubisco protein is particularly important, right? And that will also be something, if you mess with Rubisco, and which is one of the main proteins that lets the whole Kelvin cycle happen, right? If this cycle can't happen, then basically the whole process gets held up. That's just like not having the oxygen in cell respiration, right? So these are just things to think about. What you need for this process to go on, those are things that, those are sort of the vulnerabilities in the process. Plants can intentionally regulate their intake of CO2 for when photosynthesis, they want photosynthesis to happen and when it doesn't. Plants, and generally, organisms are not going to try to denature their own enzymes, right? But they could have, those key enzymes, those would be good places they need to regulate it. Generally, I don't think this really happens in cell respiration or photosynthesis, but in a lot of processes, those key enzymes are the ones that are going to have those activators or inhibitors, right, because those are really the key enzymes. And I think I may have mentioned, I don't know, in cell respiration, that these enzymes, proteins are everywhere. That's not super important. That's not generally how the organism itself will regulate cell respiration. Really, how the organism will, or really what I mentioned these for is, this is where the organism is going to, or not the organism, this is where you, if you're trying to disrupt this process, if there's a disruption in the process, these enzymes are places where the process is very vulnerable. Right, just want to clear that up. Okay, the last thing is photorespiration. And I don't have this diagram, but I had one where it was, there was a big smiley face here, and a big frowny face here. Right? Kelvin cycle, good. Photorespiration, bad. Right? And this is a process that they might put something like this diagram on the AP, they could. Essentially what happens is you have oxygen being fixated by Rubisco instead of carbon dioxide. And the big problem with that is when photorespiration goes on, you don't get the sugar out at the end, and you lose energy, which is not good, right? And so plants will oftentimes try to regulate photosynthesis, right? That's when a lot of the regulating where carbon comes in or what parts of the cell get the carbon dioxide, right? They want to regulate the photosynthesis and they want to regulate the Kelvin cycle to basically stop the oxygen, right? Because oxygen is produced in photosynthesis, so they don't want this oxygen to get into the Rubisco when the carbon dioxide should be. So what plants will sometimes do, they either will separate the light reactions, which produces oxygen, and the Kelvin cycle, either by space, which some reactions some cells go through the Kelvin cycle, some cells go through the light reactions, or by time. So they'll have the light reactions happen in the daytime, right? And then not let in any carbon dioxide during the day, and then open up their stomata at night, and so that the carbon dioxide comes in at night, the Kelvin cycle will go on at night, right? So that's why I'd mentioned, the opening and closing of the stomata. They can control, essentially, when the Kelvin cycle will go on by when they let the carbon dioxide in, right? And so that's one way that they actually minimize on photorespiration, is by regulating this by and letting this carbon dioxide enter at night when the light reactions are not going on and producing oxygen. So you have a lower oxygen concentration in the cell when the Calvin cycle is going on.